Bismillahirrahmanirrahim. Let's talk about chronic pancreatitis. Chronic pancreatitis is actually a very prolonged disease which results in the inflammation with irreversible tissue damage. So what is happening here is that there is prolonged inflammation resulting in irreversible destruction of parenchyma. Then there is fibrosis and there is destruction of endocrine parenchyma in the late stage of the disease. The risk factors of the chronic pancreatitis include chronic alcohol consumption, pancreatic duct obstruction. Then there is autoimmune pancreatitis, which is characterized by the IgG4 antibodies. Then there is genetic predisposition to the disease. Let's talk about the pathogenesis of chronic pancreatitis. What is happening in the chronic pancreatitis is that there is a continuous cycle of injury and healing. It results in the activation of tissue growth factors as well as platelet-derived growth factors. So these growth factors result in the activation of myofibroblasts, which increases collagen deposition inside the parenchyma. So as a result, there are two things. The first thing is that there is fibrosis, and second one is SNR cell destruction. So both of these functions are going on hand in hand, resulting in decreased functionality of the pancreas. In the late stage of the disease, the islets of Langerhans also get involved. So what is the role of alcohol in the pancreatitis? First of all, alcohol directly constricts the sphincter of Oddi. So the constriction of sphincter of Oddi results in decreased drainage of pancreatic enzymes from the parenchyma. Hence, there is a rise in the concentration of enzymes in the parenchyma. So the next is oxidative stress. Oxidative stress means there are increased production of reactive oxygen species inside the cells. So what are these reactive oxygen species? They include hydroxyl ion, superoxide ion, and hydrogen peroxide. So reactive oxygen species activate the pro-inflammatory transcription factors such as nuclear factors and AP1. So this results in increased production of cytokines which are pro-inflammatory. So this results in the inflammation of pancreas. The next is fusion of lysosomes and zymogen granules. Once the fusion of zymogens occur with the lysosomes, there are hydrolases present inside the lysosomes. So these hydrolase enzymes result in the proteolytic cleavages and produce activated enzymes. The next is that the reactive oxygen species directly damage the mitochondria. So the injury to mitochondria results in increased calcium influx inside the SNR cells. As you may recall, hypercalcemia within SNR cells of pancreas result in activation of trypsinogen into trypsin, which can damage the pancreatic parenchyma, resulting in pancreatitis. So another thing that happens in chronic alcoholics is that there is a secretion of protein-rich fluids in the ducts. So this protein-rich fluid can form the protein plugs inside the pancreatic ducts, resulting in pancreatic duct obstruction, which gives rise to pancreatitis. So the hereditary factors that are involved are PRSS1 gene, in which there is gain of function mutation. So what is happening as a result of PRSS gene 1 mutation is that the trypsin becomes resistant to negative feedback mechanism. So there is higher concentration of activated trypsin inside the parenchyma, resulting in injury. SPINK1, as you may recall, is produced by the SNR as well as the ductal cells of pancreas. SPINK1, as you may recall, inhibits the activation of trypsinogen. So once SPINK1 is mutated, it cannot function properly, resulting in increased activation of trypsin and injury. CFTR gene, cystic fibrosis transmembrane conduction gene, once there is loss of function mutation, there is decreased secretion of bicarbonate by the ductal cells. So there is higher chance of protein plugging that can obstruct the pancreatic ducts. So these genetic mutations are also linked with the increase in the chances of pancreatic carcinoma by 40%. Coming on towards the morphology, there is extensive fibrosis in the pancreatic parenchyma. There are dilated pancreatic ducts. The ductal epithelium is either hypertrophied or undergoes squamous metaplasia. You can also see there are islets of Langerhans which are damaged by the end of the disease. There are some inflammatory cells resulting in inflammation of pancreatic parenchyma. The atrophy of the SNR cells occurs definitely due to the repeated injuries. So the islets of Langerhans are lost in the advanced diseases. This is a spared islet of Langerhans which might be functioning properly. The ductal epithelium has been replaced by the squamous metaplasia, and in case of autoimmune pancreatitis, IgG4 producing plasma cells are also found in the pancreas. If you have any question, do let us know in the comment section. Thank you.